Hi everyone! In today's video, I'm going to discuss aspects of tau aggregation and how they can directly correlate to Alzheimer's disease, which you may or may not have heard about before. This is tau, a simple colorful looking protein that actually has a huge function. This small little protein is a neuron-specific, highly soluble microtubule-associated protein, also known as MAP or MAP. Tau essentially regulates microtubule stability in the central nervous system of human beings and becomes a key factor in a well-known disease that you may have heard of called Alzheimer's disease, which I will discuss today. When there isn't a disease and we're dealing with a healthy neuron, tau is vital for axonal outgrowth of neurons and synaptic plasticity that ultimately helps the brain and you as a human to function. In essence, tau has various key parts alongside an N and C terminal that proteins usually have. Its binding to microtubules in the brain is controlled by a microtubule binding region known as MTBR, as well as the proline-rich region of the C terminus of the protein, indicated right there with those large red arrows. Microtubule binding domains of tau contain a number of lysine residues as well, of which the positive charges drive tau to bind to negatively charged microtubules. Tau unfortunately doesn't always properly function. When pathologically altered, tau molecules no longer properly stabilize microtubules and can become insoluble aggregates such as neurofibril tangles or neuritic plaques. Accumulation of these abnormal tau aggregates in a neurological standpoint is signature to multiple neurodegenerative disorders, one of which is Alzheimer's disease. Alzheimer's disease is the most common type of dementia and a leading cause of death. In just the United States alone, 5.7 million individuals of all ages have Alzheimer's disease, and this number is only growing. This disease starts off with dreadfully affecting a person's memory and ability to carry out even the simplest tasks, and the victim's conditions gets worse as he or she ages. As of now, Alzheimer's is a disease that cannot be prevented, slowed, nor cured, but what can and may be inducing this disease is intensively being researched. With more and more research, researchers are studying tau's effects on Alzheimer's disease. Tau starts to form tangles inside neurons, causing detrimental effects. Due to the extremely important pathophysiological importance of tau, efforts on understanding the way tau actually aggregates and to prevent or reverse this process has been made. However, the problem is there has been a lack of suitable method for monitoring tau aggregation. This is when FRET, otherwise known as fluorescence resonance energy transfer, comes into play. This highly useful protein technology has aided in investigating tau-tau interaction in cells and to clearly identify what conformational changes occur in tau that promote aggregation. It gives a clearer picture on the movements of tau and what regions may be compacting or folding together. But how does FRET work, you say? FRET works just as its name says. In this method, there are two key components, usually two parts of a protein of interest, in which the parts are labeled with either a donor fluorophore or an acceptor fluorophore. If these components are in close proximity to one another, energy transfer in the form of fluorescence occurs. In other words, the intensity of fluorescent color of the first donor region of the protein transfers its intensity to an acceptor, making this region glow brighter. Thus, there's a subsequent reduction of fluorescence intensity of the donor and an increase of the acceptor. The results of FRET is demonstrated on our FRET efficiency graph. In this example, FRET was used under normal buffer conditions with tubulin absent in solution in the first part of the experiment, and then the second part, tubulin protein which forms microtubules, was added. Although the details of this particular experiment aren't important for now, it is important to notice that the dotted line in the middle of the graph is used to indicate the difference and shift of the peak between the two graphs. If there is a high FRET efficiency value in which the peak is shifted right, this means that the protein has gone from a low to high compaction or has folded. This is vice versa if the peak has shifted left. Essentially, by comparing the differences of these efficiency values, FRET reveals conformational changes of proteins from their genuine structure. So how has FRET been used on tau? In one particular experiment, researchers Shauna Elbaum-Garfinkel and Elizabeth Rhodes used the method of FRET to study the energy transfer that can potentially go on in tau aggregation in the brain. The tau protein was first studied with FRET under normal buffer conditions and then analyzed again with FRET under the same buffer conditions and an anionic aggregation inducer shown here. This anionic cofactor called heparin fundamentally acts to facilitate and progress tau aggregation in a studied environment. Heparin increases solvent exposure of the aggregation initiating components of tau, increasing the probability of contact between these regions so that they could be observed using FRET. Through their experiment, Elbaum, Garfinkel, and Rhodes deduced that there were conformational changes of the tau protein. Normally, tau has a sort of S conformation in which the N terminus and C terminus are folded in S formation as shown in the faded portion of the tau protein. However, when tau began to aggregate, the N and C termini lost interaction and essentially flopped out, giving a less set structure, which is demonstrated in this more opaque structure of tau. In addition, through looking at the FRET efficiencies of the protein after placing the donor and acceptor fluorophores on the MTBR and proline-rich regions of tau, it was found that these were the regions that began to compact during aggregation. On top of the loss of interaction between the two termini, this effectively enhanced aggregation for tangles to form. And so we have it. Is this a solution to a common brain disease amongst many individuals? No. But it's a step in understanding a factor of Alzheimer's disease and what can potentially happen inside a human brain. The future of tau and aggregation only has yet to get a full kickstart. Possible therapeutic strategies such as using drugs to reduce C-termini interaction with the MTBR region or even using saline solutions to reverse or halt the spreading of tau pathology are hopes to aid in Alzheimer's tau and effectively decrease the 5.7 million number of individuals who are affected on an annual basis.